Chapter 15, Lecture number 6, the innate immunity. We are resistant to most plant and animal pathogens. However, we're not innate to all of them, and that is due to genetics. There are three main lines of defense: your external skin, which is the epithelial tissue, your connective tissue, and your inner organs. Tell me why. Now think about temperature. The body's first line of defense refers to the structure, chemicals, and processes that work to prevent pathogens entering the body. As mentioned earlier, it will be the skin and mucous membranes of the respiratory, digestive, urinary, and reproductive systems when we refer to the first line of defense. What is the role of the skin? Well, we have two major layers, the epidermis and the dermis. The epidermis is the outside, and it has multiple layers. And the shedding of the dead skin removes the microorganisms. In the case of the dermis, we talk about the collagen fibers. What about the skin? Well, your skin has chemical defense, and this gives us antimicrobial peptides, which are known as defensins. These are proteins. Your perspiration also secreted by the sweat glands. They generate salt, and due to the sodium chloride, it makes the growth of the pathogens very difficult. It also contains enzymes known as your lysozymes. What else do we have? The oil glands, known as your sebum. And these have a very good defense against the microorganisms because it lowers the pH of your body. What are the mucous membranes? Well, the mucous membranes are the ones that have all the body cavities open to the environment. These are known as your epithelium. And your epithelial tissue are living cells. But most important, they contain goblet and ciliate columnar cells, which help remove the invaders. Below the epithelium, you have the deeper connective layer that also produce chemicals that defend against pathogens. This is the structure of the respiratory system, which is also lined with the mucous membranes. This table gives you the first line of defense, which is the comparison between the skin and the mucous membranes. What is the lacrimal apparatus? Lacrimal means lacrima in line, which means for tears. It also contains very important lysozymes, which help us destroy the bacteria that invade. And your lacrimal glands are, as you can see, right before the eye surface. What is the role of normal microbiota in the innate? Okay, when we talk about normal microbiota, those are the organisms, microorganisms, bacteria that live in our organs, and they help us actually not only with the digestive component, but also to fight against bacteria. How so? Well, the invading bacteria have to compete, and they have to compete with the normal microbiota that we have in our organs, and therefore it keeps the immune system peaceful in good times and helps stimulate inflammatory response when bad guys are around. We also have antimicrobial peptides. Now, when we talk about peptides, of course, we're talking about the ones that make up with what? The amino acids. They're in your skin, your mucous membrane, and also in your blood vessels, okay, in the neutrophils. Table 15.2 gives the secretions and activity that contribute to the first line of defense. From your saliva, your stomach acid, bile, your peristalsis, and even the urinary system. Yeah, also the secretions and activities that contribute to the first line of defense from the reproductive system and the cardiovascular system. Tell me why. Why is this disease dangerous, even though we still lose the skin cells? What is the second line of defense? Well, that's below your first line, which is, from the skin and also from the glands and sebaceous glands. We go onto the mucous membranes now. Okay, this has antimicrobial chemicals also, and many of these components are contained or originate within the blood system. So what is your blood system? Well, first of all, plasma. It's mostly the water, okay? And most important, it also contains the iron-binding compounds, which are needed for your metabolism. What are the defensive blood cells? Your leukocytes. Those are your white blood cells. However, we also have the erythrocytes, which are important. Those are the red blood cells that carry oxygen and the carbon dioxide in the blood. We have the platelets. These are involved in blood clotting. And again, the leukocytes are the ones in the defensive mechanisms, which are classified as the granulocytes and the agranulocytes. This is a schematic representation. And as you can see, the leukocytes are the innate immunity, second line of defense, and also the adaptive immunity. And we also refer to the macrophages, which are the ones that actually engulf the bacteria. You need to make a table of the leukocytes. That will help you for your test. So we have your leukocytes, which are known as two main types, your granulocytes and your agranulocytes. In the case of granulocytes, as you saw previously, you have your basophils, your eosinophils, and your neutrophils. And in the case of the neutrophils and eosinophils, those are the ones that are your phagocytizers. In other words, they act like your amoebas. They engulf the particles, such as bacteria or other invaders, and break them down. Your granulocytes are the ones that are known as your lymphocytes and monocytes. Lymphocytes are known as your natural killers. So when we have a lab analysis and you have an increased elevated amounts of leukocytes, that means what? Well, pretty much that you have an infection. Phagocytosis. These are known as your phagocytes. Okay, they actually uh, engulf particles in 6 stages, and you're seeing right here. First is chemotaxis, and then there's the adherence component. In other words, the ingest the microbe, and then once it is ingested, they are broken down and killed, and then they are eliminated by exocytosis, which is what you're seeing right here. And these are the pseudopods. Pseudopods is false feet. So other white blood cells also do killing of the actual organisms or microbes, but they don't do it by phagocytosis. They actually secrete toxins, in other words, chemicals, and these chemicals are actually the ones that kill them. Some of them actually secrete mitochondrial DNA, and with the proteins, they form a structure that kills the bacteria, especially in the intestine. Others also secrete toxins, and they do this into the viruses, so they actually get rid of cells that get tumors. However, for some reason, the malignant tumor, which is the one that causes cancer, do escape. Again, other examples of non-phagocytic killing by the neutrophils. These generate extracellular fibers. It's sort of like a net. Well, it is a net. And then they entrap the microbe and break it down. And this generates cell death, and that's known as apoptosis. Toll-like receptors, these are non-specific chemical defenses against pathogens, and these actually bind to pathogen-associated molecular patterns, which is known as PAMPs. And once they do that, because of an infection, they actually generate apoptosis, and they also stimulate the adaptive immune response. This is a table giving you the toll-like receptors and the nitro microbial patterns, which are the PAMP. This body's second line of defense, we also have non-specific chemical defense against pathogens. These are known as NODs, nucleotide oligomerization domains. They work just like your TLRs. They actually bind to also the PAMPs in order to trigger the inflammation. Now, the inflammation, believe it or not, it's not a bad thing. It's actually it's a good mechanism that actually increases blood flow. And by increasing the blood flow, it actually increases the leukocytes' defense mechanisms. Also, what we have as non-specific chemical defense are your interferons. These are proteins. They're known as cytokines, and they're actually very important because they actually release a response to bacteria, parasites, and tumors. They're also known as alpha and beta for type 1, and type 2 are the gammas. Interferons are also very helpful against cancer and hepatitis, specifically hepatitis C. And as you can see, what the interferon does, it inhibits the actual virus from transcription, as you can see right here. And by inhibiting transcription, the virus cannot make protein, and without protein, therefore it cannot replicate. This is the type 1 and type 2 interferons and the actual inducing agent and actions. keep going with the body's second line of defense. And now we're going to serum proteins. And this actually complements the activation results in the lysis of foreign cells, meaning that it actually helps to get rid of cells that are not to your body. In other words, they're foreigners. So lysis means to break down. The actual complement can also activate in 3 ways, which is the classical, the alternative, and the lectin pathway, which is you see here. And this basically means whatever antigen or antibody is present, it will alternative go either the classical or the alternative pathway or the lectin. This basically means the activation goes by factors known as proteins. These are protein activators. And this shows you the cascade. As you can see, it is a metabolic pathway. It doesn't go with one direction. It actually triggers a multiple array of proteins. Now, don't worry about the details. All it means is that whenever you have an activator present, is because there is an invader, and therefore it makes all the actual proteins relay to one another in order to make a prediction of what is present in order to get rid of it. So when do we stop it? Well, once it is complemented, it means that your body already has the first and second line of defense present. Therefore, there are other proteins that bind and break down activated complementary proteins once the actual infectious disease is killed off. And now we're back to the inflammation. What is it? Well, it's characterized by redness, heat, swelling, and pain. It could be acute or chronic. When it is acute, it means it just develops very quickly. And again, like I mentioned earlier, it is beneficial because it is important for the second line of defense, increases the blood vessels permeability, and like I mentioned earlier, also the migration of your white blood cells. What is chronic? Well, chronic refers to long lasting, okay? And this damage can actually be very, very, very difficult to cause major disease in your organ system. And here it is. Once you have the release of inflammatory mediator, it means proteins are present, okay? And these proteins are your bradykinin, prostaglandins, your leukotrienes, and histamines, okay? And these are actually used for signs of inflammation in which your antihistamines or antiprostaglandin will be released. And as you can see in this picture, you're going to see the dilating effect of the different proteins I just mentioned to you, which are your inflammatory mediators. And this diagram depicts even better how your monocytes now can actually leak through the blood vessel because of the inflammation. And therefore, this squeezing through the vessel is known as diaperesis. And this actually will mediate and help your monocytes to get rid of the infection. And that's what it shows you, the migration of the phagocytes. It attaches to receptors via margination, which is what you saw right here. This is what refers by margination. It squeezes through, and then it attacks the infected disease. The first arrival is by the red blood cells, of course, the neutrophils, and then the monocytes. What happens through inflammation? Well, after that is your tissue repair, okay? Right after you have the inflammation, you have tissue damage, so it has to be repaired. And you get extra nutrients and oxygen, However, some of them cannot be fully repaired and they become scar tissue. And this shows you the actual stages. As you can see here, we mentioned earlier, some of them cannot be repaired. That's the accumulation of actual damaged tissue and pus. Fever. What is fever? Well, your normal body temperature, well, you growing up here in the United States is mostly known as 98 Fahrenheit, but when we talk about Celsius, it's 37 degrees. Anything above that, above 37 to 38, 40 degrees, is known as fever. The causes of fever are known as pyrogens. Okay, pyrogens induces your fever. It could be for many causes. However, the exact mechanism is not known, but it is a positive factor up to a certain point, because if you have prolonged fever, of course, it can cause death. The hypothalamus, which is part of your brain, is the one that is known as your body's thermometer. And whenever there's a problem with your infection, that's what it increases the temperature of your body in order to get rid of the infection, because your infectious microbes, most of them, as we know, the best temperature they live is 37 degrees. So when your body increases the temperature, it actually has different effects. It kills the bacteria, but at the same time, it also enhances the interferons. It also enhances the activities of your phagocytes. And this is a great summary. And that will be the end of chapter 15. Continue lecture 6. Now we're on chapter 17, which talks about immunization and the immune testing. So what is immunization? Well, we're talking about vaccination here. Okay, when we talk about vaccines, some of them are considered to be attenuated, meaning that these are viruses that could be alive, but they're modified, of course. And when we give this to the patient, it can result in a mild infection, meaning mild fever, especially to this, young babies. But it can provide contact immunity. In other words, it causes the disease to be susceptible in individuals, but only the ones that their immune system is low. Vaccine types, again, some of the ones I mentioned before are active. Now these ones are inactive. These are viruses that are dead, but when you introduce it to the body, since they contain the actual protein, they actually have the genetic makeup that your immune system will recognize when the active agent actually comes into your body. However, this become inactive after a few years, so these are the ones that you have to do every 5 to 10 years, such as the HPV vaccine, for example. The conjugate vaccines, they're not viruses, they're not bacteria, they actually are polysaccharides. In other words, they're man-made. But this polysaccharide presence in the body makes your immune system raise antibodies. Your toxoid vaccines are known as your chemically or thermally modified toxins. Okay, they actually also use to stimulate the active immune system. The combination vaccines are the ones that are partially viral and partially chemical. They also are the ones that we know as recombinant gene technology vaccines. And basically, we as the science field, we know the actual genetic makeup of a specific organism, and we do recombination DNA to replicate that actual genetic makeup. So when your immune system sees the actual organism, the microbe that is, it will recognize it immediately and boost up your immune system, which is what you're seeing right here. Vaccine manufacturing. Well, this has been happening for, especially since the 1930s, 1940s. And this happened after the discovery of Alexander Fleming. Do you remember? What did Alexander Fleming discover? So the CDC recommendation immunization schedule. Controversial. Why? Well, there has been a lot of controversy behind autism and vaccination, and the amount of vaccinations given to young toddlers. However, nothing has been very clarified about this because, for example, you might have two twins, and one of them has developed autism and the other one didn't. And if they have the exact vaccination genetic makeup, then what is the relation? And this goes into the actual vaccine safety that I just talked about. Another type of immunization now is called passive immunotherapy. Instead of vaccines, we give the patients antiserum, which actually has the ability to perform antibodies. And this has several limitations, though, because we do have patients that can trigger allergic reaction, which is known as semi sickness. And also, some individuals are not protected from these future infections after you give them serum. So, how is this limited? Well, limitations can be overcome through the development of hybridomas. Hybridomas happen to be cancerous cells, but they're not cancerous cells that affect the humans. And these cancerous cells are produced through these hybrids, and they produce antibodies that can actually be cloned and actually purified for humans. And these are known as monoclonal antibodies. And these monoclonal antibodies then are given to the patients to develop immunity. This just gives you the characteristics of immunity produced by active immunization and passive. So what is serology? This is the determination of the presence of specific antigens or antibodies in the blood serum. And these tests are useful because therefore, if we know the actual components of the antibodies, we can, as you already learned, perform tests so we can develop monoclonal antibodies for the patients. This is a useful table, which gives you the different immunological tests and the use. And one of them you might have heard about is the ELISA test, which is the one right here. It's actually the one which is mostly used in the industry. And this, which is the end of lecture 6. Have a good day.